Good morning, good afternoon, good evening. This is Dr. DeShavo, and I tried to put together an extended study guide for you. As you know, I already have videos posted to YouTube, and I kind of wanted to narrow in a little bit on some of the topics that'll be on exam one. And I tried to reorganize it for you. This is not the short study guide, this is 10 pages, but I took the list of topics from the study guide and moved it over, and I couldn't help myself. I had to put in some brief descriptions. So buckle up. This is probably going to be about a half an hour video, but if you study this, study it well, and use your book and your notes to supplement and review the material that's in here as well, it gives you most of the topics for the exam. So let's dig right in. As I said, I couldn't help myself. I had to do a brief description. So I went through the cell, realized the cell is like a little people. They are called eukaryotic. I always remembered that because EU sounds like you are eukaryotic. Cells that have a nucleus and a cell membrane. And I think that concept, I am going to draw probably a fair amount on this. So we have the cell membrane. Hold on just a second. And the nucleus. In your notes, there's several definitions of the organelles, or tiny little organs that are located in these cells. Review that. What I want to go over is a couple basic structures here. This cell membrane is what helps to regulate, that should be right on that cell membrane or that outer layer, what comes into and out of this cell. If you think about it for a second, if we poured all sorts of water into this cell, it would blow up and up and up and up and up and up and eventually burst, which can happen if you don't have the right tonicity in the body. And you're all aware of tonicity. You know, isotonic, isotonic are solutions that are close to our body chemistry in our blood, the correct balance of electrolytes and fluids and whatnot. And you could get hyper and hypotonic solutions that can change the cell shape. So the cell has to be able to maintain its proper shape, maintain what comes in and out at appropriate levels. And in order to do that, it has the ability to let very small particles pass through, like oxygen can sneak through, for example. But larger particles like proteins have to come to little channels that are in the cell membrane. They can pass through there. Okay, and I'm kind of getting into the weeds with it. What you basically need to remember is cells need to maintain a certain shape. They have to maintain a certain balance of water, electrolytes, lipids, proteins, and carbs. Can't get too much, can't get too little. It's got to be balanced. Okay, so that's the basics of it. In the nucleus, you probably remember from A&P, we have DNA. DNA stays in the nucleus. It holds our genetic code. But the other cool thing that DNA does is it holds codes for all the proteins in our body. And proteins in general make most of the structures of our body. RNA, if you remember, carries that code out to the cytoplasm. So DNA stays in the nucleus, RNA comes in and out, and it helps to create the structures of the proteins that are released from the cell and thereby create the structures of our body. Okay, so DNA, RNA. You need to know the bases for both. Okay, so put bases here. Adenine, thymine, cytosine, and guanine. I'm going to let you look those up. So I'm reviewing, but I'm not going to give you all the answers. Okay, look up the bases for DNA, look up the bases for RNA so you know them, and realize that those bases help to create the code for proteins. And we're going to get into that in a little bit more. So Some of the electrolytes that move in and out of cells are sodium and potassium. There's others, of course. There's proteins, there's chloride, etc. I just want to establish that sodium and potassium are super important. A really important or easy way to remember that is there are a couple of the electrolytes that we have in our body. Sodium and potassium. Whenever you look at an electrolyte drink, they have sodium and potassium. Okay. And sodium and potassium actually can cross the cell membrane and cause some very important functions that happen in the body. Now, the cell membrane, I find this fascinating, also helps it interact with its environment. For example, cells that replicate, so suppose this is a normal functioning cell I've drawn. Once it replicates itself and it fills the space it's in, it'll stop replicating. Now, I want you to think for just a second, what doesn't stop replicating? What cells don't stop replicating? What cells go awry and create more and more and more and expand and go out? Cancer cells. They have uncontrolled replication. So the cell membrane is not only important to keep things in and out, it's important to be able to interact with its environment, stop replicating when it's supposed to, and not go where it's not supposed to. So cells metabolize just as we do. They break down structures. Okay, know this. For metabolize, somebody told me once it sounded like cannibalize. I don't know. But cells break things down to use them. For example, we will break down glucose to use it for energy. If we have protein and we're creating bigger, stronger muscles, that's called anabolism. We deposit more protein into the muscles. Those help to create bigger, stronger muscles. Okay, so those are two very important definitions for metabolism. And metabolism is simply the chemical functions that happen in our body. Now, an important chemical event that happens is the creation of ATP. That's adenosine triphosphate. You may or may not remember that mitochondria are the powerhouse of this cell. They're these really funky shaped organelles that undergo a very crazy process called the citric acid cycle. You may have learned it as a Krebs cycle to create ATP. ATP needs glucose and oxygen to create it. For example, I used to think my youngest son never slept for 2 years. And I would literally eat a power bar and breathe oxygen, thinking, this has got to give me some energy for my mitochondria. I ingested the food, I breathed in the oxygen. Those together went down to my mitochondria and created these little pockets of energy called ATP. I'm trying to draw ATP. Well, you get the idea, okay? ATP is stored until we need to use it. We release, you may remember this from ANP, which we don't need to get into the weeds with this one, a phosphate. It turns into ADP, and then we could regenerate it back to ATP. It kind of goes back and forth, but you could just remember it as ATP. ATP are the little packets of energy that mitochondria create. Now, in the mitochondria, and this is so much more complex than I'm even giving you, so I want to give you the general gist of this. ATP is created, once again, in the mitochondria by a process called the citric acid cycle or the Krebs cycle, okay? Crazy, crazy complicated. I just want you to understand the concepts. So we can create it without oxygen and glucose, but it's way more efficient if we have oxygen and glucose, okay? So oxygen and glucose go into the mitochondria, goes through this cycle. You can look it up on your own, look at all the steps, and you're welcome for not having to memorize all of them. It's crazy, okay? In that citric acid cycle, there are several steps that turn out a bunch of ATP. So glycolysis is the first step. That occurs outside of the mitochondria. There's several steps in that. You just need to know glycolysis. And then oxidative phosphorylation, which occurs in the mitochondria, will create a lot of ATP. It's so complex. Just know these very, 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 very basics. Okay. So if we don't have enough glucose, a lot of people are on the keto diet, right? And they use fats. And the whole gist behind that keto diet is that we'll start to use fats for energy. That's great. We can create energy in the citric acid cycle when we feed lipids into it, but we have to actually chemically turn it back into glucose. So we use energy to make energy kind of thing. And if we pile fats into there, into that citric acid cycle, which is in the mitochondria, we'll get half of the ATP. So yes, we're creating energy, but we're getting half the ATP. I know that people, once they're on and this is just a side note, if you're interested, they're on the diet, they feel like they have more energy eventually. And I'm not quite sure how that happens. Maybe you're burning more fat and creating more energy would be my guess. So anyway, lipids would be, also known as fats, your second option for your energy in there. Your last, and of course, minimal option, you do not want this, is protein. This is when we start to see people waste. People who are anorexic, we see this, if you've worked in a nursing home. My father-in-law is in a nursing home right now. They're all so thin because they don't eat. God love them. They start to use their muscle for energy, and that barely produces any ATP at all. And you're starting to undergo what's known as wasting when a patient starts to get thinner and smaller and more frail. So if there's no oxygen, we can create energy. And once again, this creates very little, and it's known as anaerobic glycolysis, and the byproduct is lactic acid. I guarantee you've had this happen. You've gone out, you're going on a new exercise regimen, you run 5 miles, you get up in the morning. This just happened to me. I was doing squats, and I was like, I cannot walk, right? And that is because those muscles squeeze so hard, they had no oxygen. We created energy, but one of the byproducts of creating the energy in an anaerobic environment is lactic acid that deposits into your muscles. You get that soreness. Okay. So we know that substances move in and out of the cells. I talked to you about that. A lot of them go through those channels that I drew in that plasma membrane. Okay. Now, some of those membranes, of those channels use ATP. So that is called an active transport because it uses energy. Some are passive. They don't use energy. They just filter in and out. So, for example, diffusion and osmosis are two examples of that. Review those. Review what they are. Diffusion is basically this. Well, let's define a solution first. So if we have a beaker, we'll be all scientifical, full of salt water, okay? That salt water is a solution. If the red dots represent salt in the water, the salt is a solute, the solvent is the water. In our bodies, basically the only solvent that we're going to talk about is water. I always remember this because V is shaped like a W for water. So stupid, I know, but it helps me to remember. So the solute is a substance that's dissolved in the solvent, and that gives us a solution. And if you think about it, we have a whole bunch of solutions in our body, right? So solutes in our body could be fats, proteins, salts, potassiums, electrolytes. Okay. Just know that balance because it'll help you to understand some of the following definitions. Diffusion. Diffusion is the spreading out of the solute through the solution. I always think of it like a party. If everybody comes into the entrance of the party and there's a huge room, they will diffuse throughout the room and eventually spread out relatively evenly. And that happens with solute and solvent because of the movement of those particles. They'll eventually space themselves out fairly evenly. Osmosis is the movement of the water or the solvent. You may have done this experiment. It's called a YouTube. Not like the videos. Okay. There's literally a beaker that's shaped like this. And if you put a membrane in that's only permeable to water, and you have a ton more solute, I'm having trouble again, on one side than the other. Let's just do 2 dots over here. The water will distribute evenly to balance the solution on both sides and actually go higher, even against gravity, on the side with more solute. Okay, so osmosis is the movement of the solvent or water. Diffusion is the distribution of that solute. Now, cell functions. Phagocytosis is when cells ingest substances, and we ingest substances for a multitude of reasons. Cells break down, they wear out. If we have tissues that we can regenerate, we've got to create new cells. Phagocytosis helps to eat up the dead cells. Phagocytosis also kicks in if we have some sort of antigen that enters our body. Exocytosis and endocytosis, these are all active. By the way, they all use energy. Exocytosis, I always think of as spitting things out. Endocytosis, I think of sucking things into cells, into and out of cells. So endocytosis is, I should say phagocytosis is a process of endocytosis, but endocytosis is just bringing things in, whereas phagocytosis is when they eat things up. Phage means to eat, P-H-A-G, okay? So if we're talking about bringing things into cells to be either digested or destroyed, it's phagocytosis. Now, okay, don't blow your mind about this, okay? This is a very complex process. We're almost out of the really complicated cellular stuff, I swear. This is what I want you to know. Your muscles and nerves work by electrical impulses, very similar to electricity. Sodium and potassium are electrolytes that I spoke about. They're ions that we have in our body that are charged particles that move across muscle and nerve membranes to make muscles and nerves work. This is almost as far as we're going to go with it. Okay, so don't get too nervous. I can feel it. I can feel you get nervous. Don't get nervous. Okay, so sodium and potassium are charged particles that cross muscle and nerve membranes. They do cross other cell membranes as well. Let's just focus on muscles and nerves to make muscles and nerves work. The movement of sodium and potassium is called an action potential. Okay, and this action potential causes electrical movement to move down a muscle cell or down a nerve cell to send a signal to make them work. Okay. There are 3 steps to an action potential. Let me actually pull this up. And once again, we don't need to get too crazy into this. Okay. So here, for example, is a nerve. An action potential would move down the length of the nerve to send a signal, say, from your brain out to your hand, to tell your hand to move. That is an action potential. And that process happens with the movement of sodium and potassium across those cell membranes. And it is a very complicated process I really don't want to get into. Here is the basic structure, okay? And you can see that there's voltage measured here, time here. The nerve, if we were to actually take a nerve and put 2 electrodes across it, this is what we would see printed out on the paper. Let me clarify, this is not an ECG or an EKG. Completely different. They just look similar. So don't get those 2 mixed up, okay? When the nerve gets stimulated, it starts to have more of a positive charge. It'll peak when it's done with its action, its movement, its function. It undergoes regulation back to a more negative resting charge kind of overshoots that and then goes back to its resting state. That's all it is. Don't make it more than it is. Okay, so here's our resting nerve or muscle. Oops, we want it to work. It undergoes what's called depolarization, where it becomes more positive. It's done with its action or its nerve function, and it undergoes repolarization to regulate it back to its resting state. That is the very basic gist of it. If you don't get it, shoot me an email. Okay, and these action potentials happen all the time. They're happening in your brain right now, going nuts, saying, oh my gosh, I can't believe I have to memorize this. No, I'm just kidding. Okay, that's all you need to know. Action potentials make muscles and nerves work. It undergoes depolarization, where it becomes more positive, and we actually have sodium moving into the cell, making the cell more positive. I don't even think you really need to go that far. You can if you want. Okay, repolarization, where it becomes more negative, and those positive potassium ions are moving out. If you want to memorize that. Okay. And then hyperpolarization. Potassium is just a little slow to stop that regulation. Okay. And it overshoots. Then it goes back to that resting cell membrane. That's a tough one. These are the very, very basics. Let me know if it doesn't make sense. Mitosis, you should absolutely know. It's cell division. Okay. Not all cells divide. For example, nerves. Nerves do not create new nerve cells. Skin undergoes mitosis all the time. So think about tissues that regenerate themselves. They are undergoing mitosis all the time. Please know the different phases of mitosis. I actually sort of went through them a little bit further in the notes. So we'll look at that in just a second. Understand the basics of meiosis. This is the division of sperm and oocytes or eggs. Understand it must have half the chromosomal number. So when they join, they have the proper 23 pairs, also known as 46 chromosomes. Look at that for a second. People always get this wrong. 23 pairs, so 23 pairs, there's 2 times 2 is 46 chromosomes. It's not 46 pairs, it's not 23 chromosomes in a normal cell, okay? We're talking about normal cells here. In sperm and eggs, there are 23 chromosomes. When they join together to form any other cell, they multiply by 2 and you get 46 chromosomes, okay? The chromosomes are organized in pairs. This is where people get confused, okay? So in a normal cell, after a sperm and egg combine, you have 23 pairs now, or 46 chromosomes total. People get that confused. Please review it. Let me know if you don't understand it. All right, I am back. We had a little children invasion there. So I'm sure you all understand. You have families. So moving on to tissues. So tissues by definition are multiple cells that have a similar function. Okay. And the different shapes, and we're not going to get too much into that, of the cell helps to denote its function. And I'll give you a little example in a second. So just wanted to make sure we finish that nerve. In general, when we start to talk about tissues, we'll talk about nerve, which is nerves. Epithelial are closely packed cells that line the surface of the body. If you take your fingers right now and touch your skin, you're touching what's called epithelium. A lot of cancers grow from epithelial cells. So it's good for you to have a basic working knowledge of epithelial cells. We also see closely packed cells that are in some of our endocrine glands. And we'll look at endocrine when we get to that chapter. We'll look at it more in depth. There's connective tissue. It makes sense. Hallelujah. It connects. Okay. And then muscle, which moves. We have 3 types of muscle that move, though. We have cardiac. We have skeletal. I want you to think for a second about the third. You can even pause the tape if you want to. Smooth. Smooth is located around blood vessels. It is in the hollow organs of the body. and it helps to move substances through those hollow spaces in our body. And having a good working knowledge of those tissues helps us to understand disease processes. Now, if you think, in the prime example that everybody teaches in anatomy and physiology about the structure of cells, an important example is pseudostratified ciliated columnar. Ha ha ha. Pseudostratified means it looks falsely layered. Columnar, it means they are column shaped and they have cilia on the top. Everybody remembers that. And the cilia helps to keep the junk out of the lungs. So if I were to take a stab at drawing them, columnar means they're taller than they are wide. Cilia means they have hair-like projections on the top. There's many more than that. And those cilia have actually very small muscles that help them to move substances in one direction. So if these are our lungs here, this is our mouth here. I cannot draw, just so you guys know, I'm a horrible drawer. The pseudostratified ciliated columnar, let's just say ciliated cells because really want to focus on that, helps to push the gunk out of the lungs. We don't want stuff to get into the lungs. We all know that. The alveoli are only one cell thick, the lining of them. If we get stuff into the alveoli, our lungs could collapse. So we have the cilia to keep stuff out. People who smoke lose the cilia. So what do? They have to cough to bring that mucus up, and they get that really nasty, phlegmy cough, and they have to generally kind of spit stuff out. In normal functioning bodies, the cilia moves it up little by little, and it'll actually go down into your stomach. We digest it and we get rid of it. Pretty cool. So understanding the basics of cell structures will help you to understand the function of that tissue. Now, in a tissue, we have multiple cells, similar cells that function the same. If we have two or more tissues, we have an organ. Okay, and an example of tissues I said was the top layer of the skin. We have multiple cells stacked on each other to provide a protective layer. In an organ, we'll have multiple tissues that work together to perform a specific function. Just like your skin, you have a top layer, which you may remember is called the epidermis, and a bottom layer, which is called the hypodermis. There's the dermis as well, but just to give you two examples. And in the bottom layer, we have that fat collection. That fat provides warmth and cushioning to our bodies. It also is our storage for fat for energy. So should we run out of carbohydrates or sugars or glucose, as we spoke about earlier, we can tap into fat as a backup reserve. Cells and tissues have different abilities to regenerate and renew. For example, research has shown the liver regenerates about every 7 years, give or take. Okay, nerve cells do not. They do not undergo what's called mitosis, that replication of cells. But peripheral nerves can repair themselves. If you take a knife and you cut a nerve in your peripheral nervous system, let's just say out in the torso, arms, or legs for now, but not spine and brain, if you bring the ends of those nerves together, they can regenerate themselves. Central nervous system cannot. That is why when the central nervous system, either the brain or the spinal cord, is damaged, there is generally permanent paralysis, loss of bowel, loss of bladder, loss of sensation, loss of motor. Okay. So some damage is reversible, some is not. Some become pathologic or diseased. Okay. Now, this kind of all excludes stem cells, and stem cells are these nondescript cells that can turn into any type of cells. They used to harvest these from fetuses, but we are finding now that we can find stem cells in the body. And this is one of the premises is behind that PRP injections, is they can get stem cells and use those to modify them or push them to turn into the cells that they want. It's pretty cool. Okay. So neural stem cells are one example of those stem cells that can divide and turn into nervous system cells. Apoptosis is programmed cell death. It probably rings a bell from A&P. We also have multiple ways that cells adapt. I'm not going to sit and read through these. I'm going to let you kind of read through them and review them. I've given you a bunch of tips and ideas because some of them are tricky. I'll tell you the most tricky one is metaplasia and dysplasia. Metaplasia is reversible, so is dysplasia. Dysplasia probably sounds familiar from cervical dysplasia, from pap smears. Metaplasia is when one cell type is replaced by another. And then dysplasia is a change in that cell that reverts it back to sort of almost an original tissue. Okay, so metaplasia changes to another cell type. Dysplasia doesn't, it changes to a different form. Okay, and both of them are reversible. So we can have damage to cells too. Starting to learn prefixes is a great idea at this point. Example, hypo is beneath or below, hypoxia is low oxygen, A is a prefix that means without, anoxia is a total lack of oxygen. Okay, so I'm going to have you guys on your own go through and define these cell injury mechanisms. I've added some extras that I think are important. I think it's important for you to know ischemia, so therefore you can understand ischemia reperfusion. Review unintentional, intentional, and all of these. Any of these you want to review and define, use your book and your notes. Yeah, okay, I think the rest of that you could review on your own. Infection, okay, we're going to start to talk about infections and inflammations. It can enter the body by multiple means. And it can lead to what's called necrosis or tissue death. There are different types of tissue deaths. Please go through and define these. I've given you ones that are typically linked to certain diseases. Liquefactive generally occurs in the brain. Caseous is always linked with tuberculosis. Not to say it can't happen in other diseases, but every book that I've seen, hopefully your boards ask you this. I would be pleased as punch. Email me if it's on the boards. I know this is something that our boards liked. Hopefully, maybe it's something your boards liked. And it's caseous because it's cheesy. Oh, so gross. Okay, so look at that and review it. Gangrenous is is death of tissue with severe hypoxia. Know that as well. Woo, okay. If you need a breather, we're almost halfway through. Take a breather. But we're getting into fluids and electrolytes and acids and bases. So this is a whole chunk of information. So they're also very critical. These are going to be bread and butter. Hopefully you already have a good basis of this. And the thing I sort of did in this introduction is mind-blowing. It's mind-blowing that all these things happen all the time to balance fluids, to balance or acid base, to balance electrolytes, and we have such a narrow window of critical ranges here. So for fluids and electrolytes, it's balancing those fluids and those substances to isotonic solutions. Unless there's an issue, of course, you will treat with a different type of solution. But in a perfect world, when we are balanced, we will have the proper isotonic amount of substances in our body. So you have your waters, you have your electrolytes, such as your sodiums and your potassiums we spoke about. Chloride, we'll talk about, your book talks about briefly. I don't think I have that in the notes. And you need to keep that balanced. Okay. We also need the balance. So let me bring up this concept. So we talked about the balance in this cell. Remember, I told you we have to have that nice isotonic solution for cells where they could expand or they could shrink as well. Not only do we need to keep the fluid balance inside and outside of the cells, we need to keep it balanced from the bloodstream. So suppose this is a capillary. In capillary beds, I'm just going to put cap, okay? Capillary beds are where we exchange fluids, oxygen, electrolytes, help to balance acid bases. Capillaries are only one cell thick, So it's an easy location to transmit those fluids, and capillary beds are everywhere. Okay. We need to keep the proper balance of fluid in the bloodstream and out of the bloodstream. Okay. We need to have some fluids that continuously go through that cardiovascular circuit, and we need to have a certain amount of fluids that are in the tissues to help bring nutrients out to the tissues and bring wastes away. Okay. So this is a constant balancing act. So suppose we blow out a ton of fluid from our capillary beds into our tissues. We get edema. Okay. So just something to think about. What if we don't have enough pressure in our blood system? Okay. Remember, we have our heart. Our heart goes to our aorta. Okay. Arteries go away. Our aorta breaks down into capillary beds where we have this transition of substances. Okay. And then these capillary beds go back to veins, which go back to the heart. And I want you to look at this for a second because it basically becomes a closed loop. Okay. And the amount of blood that we have in the heart, arteries, well, realize it'll vary from arteries to capillary beds to veins, but let's think of it as a whole circuit. We've got to keep a certain amount of pressure in the cardiovascular circuit, maintaining our blood pressure. If that pressure goes too low, we cannot push fluids out into the tissues and we lose perfusion. That's a concept of perfusion. Okay. So you have somebody who comes in, they're very, they have a bleed. Okay. So they don't have enough blood in that cardiovascular circuit. They cannot perfuse their tissues. They cannot get blood perfusion through their kidneys, through their lungs, into their tissues. Okay, so that's an important concept to understand. If you have somebody who's pushing too many fluids out, there's some sort of heart damage. The heart isn't pushing blood out appropriately. It's backing up into the tissues. You can get edema. Okay, so we've got to keep the proper balance of blood pressure, perfusion, and what I just did there, and proper balance of fluid in that cardiovascular circuit and out into the tissues. Okay. All right, and then we've got to balance the fluid inside and outside of the cells. As the base, it's so important. Okay, so 3 main things to think about with acid base. Okay, the lungs. The basic thing to think about is carbon dioxide is acidic. So in the lungs, you either retain too much carbon dioxide or you expel too much carbon dioxide. With these concepts, I highly recommend you pause after everything and think about it. Okay. So I'm breathing normally. I'm balancing my pH. I don't need to change anything. I start breathing too much. I'm going to blow off too much CO2. CO2, once again, is acidic. My body will become more basic. I am a spoiled brat. I decided I'm going to hold my breath. And I hold it and I hold it and I retain too much CO2. And by the way, you know, kids say, I'm going to hold my breath. The worst that'll happen, they'll pass out. So don't worry about it. Well, I mean, you should worry about it, but you know what I'm saying. Okay. If you retain too much CO2, you become acidic. So we're thinking about our body's normal pH, which is 7.35, and the pH scale, you should be familiar with it. I'm not going to review that. If you're not, pause the tape right now and review pH scale. Okay. So our normal pH in the human body, and this is interstitial tissues, bloodstream, etc. And you might see a little variation in this number, but it should be 735 to 745. That should look familiar. What I should do is I should type these things out for you. Okay, 735, 745. Okay, so if you breathe out too much CO2, you'll go more basic. Okay, and don't forget basic is higher on that pH scale. If you retain too much CO2, you will become more acidic. Okay, CO2 is acidic, so think of it that way. Now, kidneys help to regulate acid base. They regulate it by hydrogen, okay, that's a basic ion, H plus, and bicarb. So I always think of it as like bicarbonate, which is more basic. And bicarb is HCO3. I have that further down. I have it further down in the notes. Okay. So lungs and kidneys, big contributors. Okay. Now they can also compensate for each other. And there's a third contributor. Okay, here we go. It's under here. It's a buffer system in the blood, okay? In the blood itself, it's called carbonic acid bicarb system, okay? It can push us if we are getting too basic. Hold on just a second. I'm going to pause the tape and pull up a picture. Okay, I pulled it up here. This is the bicarbonate ion. This is carbonic acid. And this is carbon dioxide. Carbonic acid is a weak acid. Hydrogen and CO2 are a stronger acid. So if we have too much CO2, we can actually bind it up as carbonic acid. It will be less acidic. Same thing can happen if we have too much hydrogen. It'll bind it up as carbonic acid because it's less acidic. It'll have less of an effect on our pH and create less acidity in the body. This happens in the blood, okay? So that's yet another way that our body can buffer our pH. So let's go back to the notes. There are 3 ways. Lungs, carbon dioxide. Kidneys, we have hydrogen and bicarb. And the buffer system is that carbonic acid bicarb system that we can tie up either hydrogen or carbon dioxide into a less acidic acid, which is carbonic acid. It's a little bit confusing. Think of carbonic acid as being the lesser of 2 evils. It's less acidic, okay? Okay, you will need to know lab values, okay? No normal range of pH. And what I mean by interstitial tissues, it's not in, say, the stomach. The stomach has a pH of 2. It's extremely acidic. Look up the normal values for that. Look up the normal values of CO2. Look up the normal values for bicarb, okay? I would also practice what happens if you have somebody go into metabolic acidosis. So metabolic generally references the kidneys. Okay. So, but it could be diabetes with ketoacidosis, for example. Okay. That references that metabolic. Respiratory references CO2. Okay. So I would practice, if I were you, not only what happens with these lab values if someone were to have some sort of kidney damage and go into metabolic acidosis, what's a compensation? A great example is ketoacidosis. Somebody goes into ketoacidosis who's diabetic, and that process we'll look at in the future, will actually start to expire more CO2. So they will have compensatory respiratory function where they'll breathe off CO2 and they'll actually go into respiratory alkalosis because they're getting rid of so much CO2, but they're trying to balance their overall pH. Okay, so I would review, diseases, compensation. You'll need to know this in the future. Okay. So know and define the following. We touched upon this a little bit. Hydrostatic pressure of the capillaries pushes that water out. Oncotic pressure means there is more solute. Okay, so if you have more solute in a substance, you'll draw more water into there. Okay, so if you have a higher oncotic pressure in the capillaries, you'll draw substances back in. Okay. And proteins are a huge factor with that. We have 2 proteins that the liver makes, and they help to retain water in the bloodstream to balance out our blood pressure and keep it at a pertinent or an appropriate pressure. Capillaries have to be permeable. This is where we have substances cross from the bloodstream into the tissues. Review hypovolemic shock, signs, symptoms, what would happen, what would cause it. What do allergic reactions happen or cause with hypovolemic shock? Why would that happen? I want you to think about that a little bit. Edema. We've reviewed sodium and potassium. ADH. Look at that substance, where it's created, what it does. I gave you some basics. Also, review SIADH, symptoms of inappropriate ADH. What causes it? How does it manifest? Dehydration, its effect and or causes with young and old. Hypernatremia, its effect on the body. I help it. Acid-base balance, compensation, buffers of the body. Okay. Know the values for acid-base. Look them up. Know normals. Okay. Know metabolic versus respiratory acidosis and alkalosis. Now, moving on to chapter 2, genes and genetic diseases. Here are some helpful links. You might want to review those. You can also use them in correlation with the lists I've given you below. I think at this point, let me see how much more. Yes, I'm going to restart at a second review since this video is getting a little bit long, and we will pick up right here. Thank you and see you in the next video.